Hello students, in this video we will be discussing in general sexual mode of reproduction and also sexual reproduction in plants. First let us examine the definition for sexual mode of reproduction. It involves two parents, formation of gametes, and the offsprings are not identical to the parents. Then what is the significance of sexual mode of reproduction? It is important because variations are created and variations help in the survival of the species over time. In sexual mode of reproduction, there is the combination of DNA between two individuals during reproduction. This may create a problem because whenever there is a combination of DNA in each generation between two pre-existing individuals, the DNA becomes double in each of the newly formed generation. And if this is the case, it will upset the control of the cellular apparatus by the DNA. And if this continues, the whole of the earth will be filled with DNA and there will be no space for anything else. Let us take the case of human beings to understand this in a better way. Human beings have 46 chromosomes and in other words we can say 23 pairs. Suppose there is a combination of DNA from two different individuals, 46 plus 46, it will result in the formation of 92. And in the next generation, it will lead to 184. Each time a new generation is formed, the DNA doubles. This, if it continues, it will completely upset the cellular apparatus. This cannot happen. So, what is the solution? Human body and especially higher order organisms have evolved a solution for this. They have a special lineage of cells which are known as germ cells, and these germ cells are found in specialized organs called sexual organs. In human body we have two types of cells, that is the somatic cells and the germ cells. Somatic cells have the full content of DNA and they are responsible for the growth and development, especially the general growth and development of the body. While the germ cells contain only half the number of chromosomes or half the content of DNA. So, when there is a fusion between the male and the female gamete, the original number of chromosomes and the content of DNA is re-established. Thus, this problem of DNA becoming double is solved. Now, there is a big difference between the male and female gamete in human beings. But there are lower order organisms where the gametes are almost similar, and in certain organisms there is only a very slight difference. Now let us see what are the differences between the male and female gamete. Male gamete is known as sperm. It is motile, it is smaller in size and does not store food. While the female gamete is larger in size, it is non-motile and it stores food. Next, let us discuss sexual reproduction in plants. The reproductive part of a plant is the flower. The flower has four main parts, that is the calyx, corolla, androecium and the gynoecium. The calyx is the outermost whorl. The individual units are sepals. They are green in color and their function is to protect the flowers in the bud stage. Inner to the calyx is the corolla. Corolla, the individual units are petals. They are brightly colored and their function is to attract insects for pollination. Inner to the corolla is the male reproductive part, that is the androecium. Each unit is known as the stamen. Each stamen is made of two parts, anther and the filament. The anther contains pollen grains in which are present the male gamete. The innermost whorl is known as gynoecium or the female reproductive part. Individual units of gynoecium is the carpel or the pistil. Each pistil is made of three parts, that is the ovary, the style and the stigma. Found within the ovary are the ovules, and within the ovules are found the female gamete. The flower in which all these four whorls are present is a complete flower. For example, hibiscus. A flower in which any one of the whorl is missing is an incomplete flower. For example, the flower of paddy. A flower in which both the male and female parts, that is the androecium and the gynoecium, is present in the same flower. Such a flower is called a bisexual flower. Example, hibiscus, rose, jasmine, etc. A unisexual flower is a flower in which either the stamen or the pistil is found. Example, watermelon, papaya, etc. Now let us see what is pollination. Pollination is the transfer of pollen grains from the anther to the stigma. Pollination can be of two types: self-pollination and cross-pollination. In case of self-pollination, the pollen grains are transferred from the anther to the stigma of the same flower, or from the anther to the stigma of a different flower but same plant. Cross-pollination occurs when the transfer of pollen grains is from the anther to the stigma of a flower in a different plant. Now the agents of pollination are insects, water, wind. Example for insect pollinated flowers are hibiscus, rose, jasmine, etc. Examples for wind pollinated flowers are all the flowers belonging to grass family, and water pollinated flowers are those belonging to aquatic plants. The flowers which are wind pollinated have a feathery stigma, while those flowers which are insect pollinated, the stigma is sticky. Pollen grains usually are light and they are well protected by a thick outer covering, to prevent desiccation. So once again, what is pollination? Transfer of pollen grains from the anther to the stigma. Once pollination is over, the pollen grains are now found on the stigma. Then the pollen grains germinate and pollen tubes are found. In the pollen tube are found the tube nucleus and the generative nucleus. The tube nucleus disintegrates and the generative nucleus forms two male gametes. The pollen tube grows down through the style and reaches the ovule. The ovule in angiosperms is an 8-nucleate structure. At the micropylar end are found two synergids, and between the synergids is the egg. Opposite to the micropylar end is the chalacil end, where 3 antipodals are found, and in the center are found 2 polar nuclei. Thus, there are 8 nucleate structure, or thus 8 nuclei are present in the ovule. The 2 male gametes which are found in the pollen tube reaches the ovule, and one of the male gamete fuses with the egg and the second male gamete fuses with the 2 polar nuclei. Therefore, in angiosperms it is known as double fertilization and triple fusion. It is called double fertilization because 2 fertilizations occur, one with the egg and the second with the polar nuclei. And it is known as triple fusion because there is a fusion of 3 nuclei, that is the 2 polar and the male gamete. With that, the events of fertilization is over. Now the zygote is formed. Once the zygote is formed, it divides rapidly and form a ball of cells and become the embryo. The other floral parts, they dry and fall off. That is the calyx, the corolla, the androecium, etc. But in some cases, the sepals persist. Such sepals are known as cadaceous sepals. And example for this we can see in tomatoes, brinjal, chili, etc., where the sepals persist even in the fruits. The ovary becomes the ripened fruit. The ovule becomes the seed. These are the post-fertilization changes. With that, we have completed fertilization, and the ovule finally changes to the seed. Now we have to examine the structure of a seed. A seed which has 2 cotyledons is known as a dicot seed, while a seed which has a single cotyledon is known as a monocot seed. Let us refer to activity 8.7, where we have to study in detail the structure of a dicot seed and that of the embryo. A dicot seed has 2 cotyledons. The cotyledons are otherwise known as seed leaves, and they store food for the developing embryo. Between the cotyledons are found the embryonic axis, which is otherwise known as the tigellum. The seed is covered by an outer covering called seed coat, which has 2 integuments. That is, the outer one is known as the testa and the inner one is known as tegmen. The seed has a scar called hilum. This scar is left by the funicle. The seed was attached to the funicle at the scar called hilum, and funicle is the stalk by which the seed or the ovule is attached to the placenta. Below the hilum is a small pore called micropyle through which water enters during germination. Now the embryonic axis is attached to both the cotyledons. The point of attachment and the plumule, this region is known as epicotyle, and the region between the point of attachment of the embryonic axis to the seed and the radicle is known as hypocotyle. The plumule is the future shoot, while the radicle is the future root. Thus, we have discussed all the parts of a dicot seed and the embryo. Now, what is germination? Change of seed into seedling with appropriate conditions such as ample amount of moisture and sunlight. Thank you.